Hello, everyone. I'm Kelly Eversole, the Executive Director of the International Wheat Genome Sequencing Consortium, and I'm very pleased to be joined today by Azahara Martin from the John Innes Center, and she's going to be talking to us about a very special gene, or element, with regards to the wheat genome. But before we get to that, I'd like to give you kind of an overview of the International Wheat Genome Sequencing Consortium. So we are an international public-private consortium. We have over 3,000 members in 71 countries. We have 10 sponsors, which make it possible for us to do things like this webinar, as well as our other activities relating to workshops and coordinating activities. So I want to take this time to thank all of our sponsors for their support, and in particular point out that Kansas Wheat has been supporting us for many years now, about 18 years. So we've had great support from the growers. So our vision after we publish the reference sequence is to continue working towards enhancing breeding through an increased understanding of the molecular basis of traits and their allelic diversity. Our activities this year is we're continuing to work with some of the wheat genomic tool developers to have new tools that the community can use. And we're continuing to gather information on manual and functional annotation of the RefSec and planning for future updates of the annotation. We're also continuing our efforts to obtain platinum quality reference sequences between 8 to 12 land races that represent the full breadth of wheat diversity, and we've secured some funding now for that, and we'll be moving forward to secure the remaining funding. We also encourage anyone who is developing genome sequences to provide these in a pre-publication release under the Toronto and the protocols for access, and we can do that through the IWGSC sequence repository. And then, as I mentioned, we do these webinar series. So we have another webinar coming up in June, excuse me. And so I encourage you to go ahead and register for that. And to explain the dashboard, there is a panel or a button that you can click that says questions. Please put your questions in the Q&A panel at any time during the presentation, and we'll get to them at the end of the presentation. Please also monitor the chat panel, because you can see messages from the organizers as well as links to papers or anything else that's mentioned during the presentations. And you can already download the handouts of both my presentation as well as Azahara. And just to remind you, the webinar is recorded and will be posted on the IWGSC YouTube channel in a few days. You can subscribe so you never miss an upload. So with that, let's move to our presentation today. And again, I want to welcome Azahara Martin from the John Innes Center. And she's going to be talking to us about the Ph1, a very famous story, / ZIP4 story. Stabilization of wheat as a polyploid and preservation of grain number. So thank you very much for joining us, and I will turn it over to you right now.
Thanks. Can you see the slide in full screen?
Yes, we're good.
Yeah, perfect. Okay, hello everybody. Thanks for registering to this webinar. Many thanks, Kelly and Isabel, for the opportunity to present my work here and share the story of the Ph1 locus a locus in which I have been working for quite a while now as part of Graham Moore Lab. And I will start by saying that the Ph1 locus is a classic and mysterious locus in which discovered more than 60 years ago. It has been widely used in breeding since the 70s and studied by many different groups. So why it is so relevant? Why there has been so much interest on it? Well, we could say that without this locus, we wouldn't have the wheat that we cultivate today. Presumably, this locus appeared during wheat polyploidization, stabilizing the wheat genome during meiosis, and by doing that, allowing wheat to be the highly fertile and productive cereal that it is. However, this improved stability and fertility was obtained at a cost. Let's say that it has some collateral effect, because the presence of this locus also prevented recombination between wheats and its wild relative, reducing that way the chances of increasing the genetic diversity of polyploid wheat. And it is these reasons, wait a second. And it is this reason, the secondary effect of Ph1, why it has been traditionally studied and the reason why it has been exploited in wheat breeding. But before I go into the pH story, I'll go very briefly through the process of recombination to clarify some concepts that I will be using later. So very briefly, the process of recombination takes place during meiosis. In the case of the male meiosis, that is what I study inside the anthers. And meiosis does two very important things, and both of them rely on successful meiotic recombination. First, it keeps the chromosome number across generations. So meiosis takes a single diploid cell and turn it into 4 haploid gametes, which later will become the pollen grain. And second, it creates genetic diversity through the process of recombination. So each one of these gametes can have potentially a different allelic combination. But I will focus only on the first stage, which is called prophase, because it is here where recombination takes place. And if you are not too familiar with the process of recombination, when you think about it, you probably have in mind this kind of pictures. First, you have the two homologous chromosomes, one next to each other. Next, a crossover is formed between them. So a crossover is the site at which recombination takes place. And these two chromosomes joined together by the crossover site is what is known as a bivalent. And finally, the two chromosomes separate with a piece of DNA exchange between them. And this is true, this is what is happening, but from this first stage where recombination has not started to this second one where recombination has practically finished, a lot more is going on. And if we want to understand and modify recombination, it is at these stages in between at which we need to act. And that is why it's very important that we know how all these processes work. Then, at the end of these 5 stages, which altogether are known as prophase, the bivalent will align at the metaphase place during metaphase 1, and homologs will separate. But focusing on prophase 1, for recombination to happen, one of the things that is absolutely necessary is that homologous chromosome synapse. And I want to define clearly this concept because I will be constantly talking about it later during the talk. So synapsis is the process by which homologous chromosomes associate along their length. And this happens because there is a protein structure that is polymerizing between them, forming what is known as the synaptonemal complex. So this complex is like a kind of glue or a scaffold that keeps homologs together so recombination can happen. Now, apart from synapses, there is much more going on during this stage, as you will see later. However, how all these processes are coordinated is still poorly understood. This is a complicated process in any species that we are now starting to understand. But if we move into it, then it becomes particularly interesting for several reasons. And I know you all here know all this, but I really want to highlight it because some of these peculiarities of the wheat genome might be one of the reasons why some of the results obtained in plant models to modify recombination might not be directly transferable into wheat, such as the huge genome size, the high percentage of repetitive sequences, and more importantly, because bred wheat is a polyploid, an allohexaploid. And one of the biggest challenges, if not the biggest, faced by a new polyploid is precisely meiosis, particularly how to discriminate between the multiple related chromosomes so that only homologs recombine. And in the case of wheat, this recognition is just perfect. As you know, chromosomes belonging to the same group and genome are called homologs, while chromosomes belonging to the same group but to a different genome are called homeologs. However, during this talk, I will use the term related chromosomes to refer to the homoeologous, because I know it can be very tricky to differentiate between homologs and homeologs when speaking, or it is at least for me. So yes, even if related chromosomes are very similar, only homologs recombine as it happened in diploid species. So what you can see here in this metaphase 1 are the 42 chromosomes of wheat, where every pair of homolog is connected to each other, forming 21 bivalents. In this case, all the bivalents are what is called ring bivalents, with 2 crossover at each end of the chromosomes. And sometimes you can also find rod bivalents with only 1 crossover connecting the homologs. Okay, so moving now into the Ph1 locus, I will introduce it with a very clear example. We can cross wheat with rye and get a wheat-rye hybrid. And this hybrid will contain a haploid set of 21 wheat chromosomes and 7 rye chromosomes. So in total, 28 related chromosomes, but there are no homologs present. So at metaphase 1, as you can see, there is no crossover formation. The different genomes cannot recombine and all appear as individual chromosomes, which are called univalent. And it was in 1958 when Ernest Sears and Ralph Riley proposed that this is controlled by a dominant locus located on the long arm of chromosome 5p. And this locus was later named Ph1 for pairing homoeologous 1. So if we now make the same cross, but in the absence of Ph1, then we get something like this. Whereas you can see there's plenty of crossover formation between chromosomes that are not from the same genome. You can see ring bivalent, rod bivalent. There is also a multivalent here at the bottom, when more than two chromosomes are connected by a crossover. So yes, this is great. By eliminating pH, we can promote recombination between related chromosomes. But what's happening in wheat itself when we eliminate this locus? As you can see in this picture at the bottom, although most of the recombination still happens between homologs, there are also univalents and multivalents present, which will be enough to produce unbalanced segregation and problems with stability. So just to summarize and coming back to what I mentioned in my first slide, this locus has the very positive effect of taking care that every wheat chromosomes recombine only with its true homologous. Also very importantly, this locus is not present in any of the diploid known ancestor, and that is why it was assumed that it's appeared during wheat polyploidization, stabilizing the wheat genome during meiosis. But at the same time, it has a very negative effect on wheat breeding, because in the presence of this locus, it's extremely difficult to introduce variability from related species. So it is a quite nice and complex story, very interesting from different point of view. So how has been Ph1 used in breeding until now? In 1977, Sears obtained a Ph1 mutant in hexaploid wheat, which he named Ph1b, and this is the mutant that has been mostly used for breeding and for most of the research related to this locus. Now, this Ph1b is a deletion mutant. It has a 70 megabase deletion on the long arm of chromosome 5p. Sorry. On the long arm, but yes, of chromosome 5p. Well, actually, we showed in 2018 that it's actually 59 megabase deletion. That doesn't really matter. But what matters is that we also showed that it included almost 1,200 genes. So as you can imagine, both to understand polyploidization and also to use it for wheat breeding, it was very important to identify the gene or the genes behind this locus. However, we have to wait almost 30 years until Ph1 was defined to a smaller region. In 2006, Graham Moore Group defined the locus to a 2.5 megabase region and later to a 0.5 megabase region, which included 9 genes. In green, a cluster of CDK-like genes, which show closest homology to CDK2 in mammals. In gray, a gene identified at that moment as a storage protein gene. And in magenta, an unidentified gene that it was named HYPE3 for hypothetical gene 3. This gene was next to a segment of heterochromatin, and the whole block was a duplication from chromosome 3b that had inserted inside the middle of the CDK cluster. The SPG was ignored because it didn't seem to have anything to do with meiosis. As for HYPE3, they used RNA interference to try to silence this gene, but none of the transformed plant was viable. So this gene was kind of parked there, and there was not much they could do at that moment. There was no tilling, no CRISPR. So they focused on the cluster of CDK-like genes, which actually was a very good candidate because these kinases are central component of the cell cycle control system, and CDK2 in mammals is involved in many meiotic processes. And this was the situation when I started working with Graham's group. So for quite a while, 4 or 5 years, I was working on the Ph1 mutant, thinking that the effect that I was observing was a consequence of the CDK cluster, until one day we discovered that the gene that had been named HYPE3 had been re-annotated and was actually a homologous of the meiotic gene ZIP4. And then we got really excited because that was a really good candidate for the Ph1 deletion phenotype. And then from this moment, finally, everything worked really well. The discovery of ZIP4 happened at the perfect moment for several reasons. First, because the IWGSC reference, the genome, was finally available. So at that point, I was doing an RNA-seq in meiotic tissue, including the Ph1 mutant, and straight away, we were able to know how many genes were present in the different deletions and how many of these genes were expressed during meiosis. And the smallest deletion, the alpha megabase deletions, included 25 genes, from which only 2 were expressed during meiosis. One of them, our candidate gene, the meiotic gene ZIP4. We could verify that there are actually 4 copies of this gene, 3 in the homologous group 3, and then this 4th copy that had duplicated from chromosome 3b and inserted into the 5p chromosome. The 3 copies on group 3 were quite similar, close to 95% to the protein level, where the copy on 5p inside the Ph1 locus was a bit more different, particularly in the first third of the protein. But more importantly, it was the ZIP4 copy on 5p, the one showing the highest level of expression during meiosis. You can see the ZIP4 copy on 5p in magenta on the left, and on the right is the expression in the Ph1b mutant, where this copy is deleted. Also very interesting, the expression pattern of this copy is completely different to the copies on group 3. While the ZIP4 copies on group 3 are only expressed during meiotic tissue, the ZIP4 copy on 5p is expressed in all type of tissue, roots, stiller, leaves. Here I show a screenshot taken from the XBIP expression browser, where you can easily visualize the difference in the expression pattern of this copy. And then the second reason why this was the perfect moment to know about ZIP4 was that the tilling population in hexaploid with cadenza just became available. So I selected line with potentially interesting mutation in the ZIP4 copy, the only one with an early stop codon and several with missense mutations. And to check whether this mutant had the phenotype of the Ph1 deletion, we crossed them with Aegilops variabilis, a wild relative. So you can see here in the picture that the wild type doesn't show any crossover. Well, sorry, actually, it has a mean number of 1.5 crossover. So yes, there was a lot of crossing and scoring, all done by Lola Ray, who was in our lab for 2 years during her postdoc and did all this part of the work. And it turned out that 2 of the mutant lines crossed with Aegilops variabilis showed a great increase in the number of crossover, more than 12 crossover per cells. The one with an early stop codon and one with a missense mutation. And if you look at the picture at the bottom, I think that the result is even more obvious. So yeah, that was great. The result was absolutely clear. The ZIP4 mutant lines had the same effect on crossover than deleting the whole Ph1 locus. And finally, Just to prove it further, in collaboration with Mark and Shadia at JIC, we also use CRISPR to target exclusively the ZIP4 copy on 5p. And to make a long story short, again, it worked really well. Mark and Shadia managed to get a line where a big part of ZIP4 had been deleted, exactly 114 base pairs. And when this line was crossed with Aegilos variabilis, again, the result was absolutely clear. There was a huge increase in the number of crossover in these hybrids. So this is it. After 60 years since the discovery of Ph1, we can finally say that a single meiotic gene, ZIP4, inside the Ph1 locus is responsible for the Ph1 effect on recombination. And equally important, we have now several ZIP4 mutant lines available, with the advantage that this line have only the ZIP4 copy on 5p mutated instead of the almost 1,200 genes deleted. So yes, we have the gene, we have new lines, but how does it work? So coming back to prophase 1, I mentioned before that the synaptonemal complex is a kind of a scaffold on top of which recombination takes place. But really recombination starts earlier with the formation of double strand breaks by the enzyme ESPO11. And I don't want to get into all the processes involved because it can get a bit complicated. But I want to show you this slide so you can have an idea of where ZIP4 sits inside the whole recombination network. So putting it very simple, the cell intentionally produces these breaks, thousands of them in the case of wheat, because it is by repairing these breaks that the process of recombination can happen. Next, for a crossover formation, the breaks need to be repaired using a homologous chromosomes. And there are two crossover formation pathways, the interferent pathway on your left and the non-interferent pathway on your right. And there are also at least 3 independent anti-crossover pathways which suppress recombination in plants. And the crossover interferent pathway on your left is the main pathway in plants, responsible for about 90% of the total number of crossover. And ZIP4 is this gene here. It's one of the ZMM protein that is controlling this main pathway. Now, the exact molecular mechanism how really ZIP4 work is not known in any species, as it happened with many meiotic proteins. However, it has been recently shown in yeast that ZIP4 could be acting as a scaffold protein, facilitating physical interaction between components of the chromosome axis and the recombination machinery. So it could be acting as a hub, connecting crossover formation and synaptonemal complex assembly. And also ZIP4 mutant have been obtained in several diploid species, and they all show a huge decrease in the number of crossover, and they are mostly sterile. So what about in wheat? Well, I mentioned before that synapsis is absolutely necessary for crossover formation. So it was proposed that related chromosomes might not be able to synapse in the presence of pH. So this was the first obvious process to check. And to do it, I used the wheat-dry hybrid, because in this hybrid, remember, there are no homologs. So any synapses that we detect will be happening between related chromosomes. And to follow the process of synapses, I use antibodies against 2 of the main structural proteins of the synaptonemal complex, ASY1 in green, which labeled the lateral element of the synaptonemal complex. In which it labeled the axis of every chromosome before it has synapse, and ZIP1 in magenta, that is part of the central element of the synaptonemal complex and labels the regions that are synapse. And I will show you first the wheat-dry hybrid in the absence of the Ph1. Here you can see cells going through meiosis, which are called myocytes, are different stages of prophase, progressing from the top to the bottom. In blue is the DNA, in magenta is ZIP1, labeling the synapse regions, and in green is ASY1, labeling the chromosomes that are not synapse. So in the absence of Ph1, we were expecting to see some synapses because there are crossovers later, and as you can see in magenta, there is quite a lot of synapses. And I'll show you now the hybrids in the presence of pH, where the assumption was that related chromosomes were not allowed to synapse and there shouldn't be any ZIP1 labeling in magenta. However, as you can see, it looks just exactly the same. We also calculated the percentage of synapses, and it wasn't significantly different whether Ph1 was present or not. So this was a surprise, because contrary to what it was always suggested, synapses between related chromosomes is not prevented or even reduced in the hybrids by the presence of Ph. So then if related chromosomes can synapse whether Ph1 is present or not, why they only recombine when Ph1 is deleted? So after that, I have been trying to develop with antibodies and doing immunolabeling with many of the proteins involved in this pathway. But I won't show any of these because either they didn't work, or when they worked, I couldn't find any difference whether Ph1 is present or not. So I just want to mention one of the last proteins to be loaded in this pathway, the mismatch repair protein MLH1. Because in all diploid plant species, the number of MLH1 sites at diplotin is the same as the number of crossover. So MLH1 is considered as a cytological marker for interference crossover. However, again, the number of MLH1 sites in green in the images was the same in the presence and absence of the Ph1. While in the presence of Ph, there is none or one crossover, and in the absence, there is between 7 or 8 crossover. So this result was really shocking, because the number of MLH1 sites doesn't correspond to the number of crossover, and this contradicts what it has been observed in other species. But then these other meiotic studies are normally done in diploid species, where they don't need to deal with all this problem between homologs and related chromosomes. So what we think might be happening is that when MLH1 is loaded on synapse-related chromosome instead of homologs, for some reason, the MLH1 site can't be processed further as a crossover. So it seems that there is a kind of checkpoint here in which to avoid crossover between related chromosomes. However, in the absence of Ph, some of these MLH1 sites manage to go further and crossover, which indicates that Ph1 deletion can alleviate to some extent these MLH1 sites stalling on related chromosomes. So based on these results and the results on synapses, it seems that at least in the hybrids, Ph1 doesn't have a major effect on early meiotic events, and everything is the same whether Ph1 is present or not. However, there was still another possibility, which is that even if the amount of synapses was the same, the timing was different. So I decided to follow the timing of synapses in relation to the telomeres dynamic, because in many species, including wheat, the process of synapses starts from the end of the chromosomes, while the telomeres cluster at one pole of the nucleus, forming what is known as a telomere bouquet. This bouquet persists for a while until quite a lot of synapses has happened, and then the telomeres disperse. So I combined the immunolabeling of ASY1 in green, ZIP1 in magenta, and the labeling of the telomeres in red. And then we realized something really interesting, which is that in the wheat-dry hybrids, independently of Ph1, there was no synapses during the telomere bouquet. It was only later, when telomeres were dispersing, when synapses could happen, which indicate that related chromosomes cannot synapse during the telomere bouquet. Only homologous chromosomes can. So this was a very significant result, because this suggests that synapsis is a highly specific process during early meiosis, but later, after bouquet dispersal, the high requirement for homology is eliminated, and synapsis can happen between chromosomes that are not the same. So this was really a key observation. Now, this is in the wheat-dry hybrids, but what about in wheat itself, where there are both homologs and related chromosomes present? Well, at the telomere bouquet, as it was expected, there is already quite a lot of synapses. However, after bouquet dispersal, synapsis seems to be delayed in the absence of the Ph1. In the presence of Ph, on the right, at this stage, synapsis is almost finished. Remember that in green, you can see the chromosomes that are not synapsed. While in the absence of Ph1, there is still many chromosome segments that are not synapsed. There is much more green labeling. And if, as I said before, synapses at this later stage can happen between chromosomes that are not homologous, then these incorrect synapses allowed in the absence of Ph1 would explain the low level of multivalence and univalence present at metaphase 1 in wheat itself. So putting everything together, the main conclusion from these cytological studies is that first, whether or not Ph1 is present, synapses between related chromosomes is not allowed during the telomere bouquet, which suggests that with polyploidization didn't require major changes in the meiotic program. It already had a mechanism in place to sort homologs from related chromosomes. So then the meiotic adaptation observed in hexaploid wheat probably just needed some fine-tuning, some improvement in the recombination machinery, which Ph1, or we can say now ZIP4, did by first making sure that all homologous synapse early during meiosis, and second, in the cases where there was still some synapses between related chromosomes by preventing sites on these chromosomes from becoming crossover. So consistent with the observation in yeast, ZIP4 in wheat seems to have an effect on both synapses and crossover formation. Okay, now I will pass to the second part of the webinar, where I will talk a bit more about fertility and preservation of grain number by the ZIP4 copy. And I mentioned earlier that in wheat itself, in the absence of pH or the duplicated ZIP4 copy, just over half of the myocytes showed abnormalities. However, the rest of the myocytes look perfectly normal, so one could think that maybe only the balanced gametes pass to the next generation with no major effect on fertility. However, that is definitely not the case, and I'll show you here a couple of examples. On the top, you can see a heat map comparing transcription in wild-type wheat and in the Ph1b mutant. So in dark blue, you can see the genes only expressed in wheat with no coverage in the mutant, and in orange, the ones more expressed in the mutant. So we can clearly visualize the 59 megabase deletion in Ph1b. But we also can detect many other deletions and chromosomes reorganizations. And then another example is this octoploid triticale that I synthesized in both the present and absence of the Ph1 locus. On the left, you can see triticale wild type, where sometimes there was some aneuploidy. This is quite usual, but nothing compared with the two examples of triticale plants in the absence of pH on the right. These were a complete mess with plenty of reorganizations and recombination between related chromosomes, as you can see, indicated by plenty of arrows. And of course, most of them were sterile. So as I said, yes, definitely some of the unstable gametes pass to the next generation and they produce fertility problems. So we decided to explore this a bit further. And to do it, we used the CRISPR ZIP4 mutant that we had generated. So in this mutant, as in the Ph1b, 56% of the myocytes present univalent or multivalent at metaphase 1. So the idea was to follow the progression of meiosis until the formation of the mature pollen grain to check the direct effect of ZIP4 in pollen formation and fertility. So here you can see the wild type on the top and the CRISPR mutant at the bottom. And keeping it short, during anaphase 1 in the CRISPR mutant, there were frequent lagging chromosomes, split sister chromatids, chromosome fragmentation, and as a consequence, the tetrads containing the 4 haploid gametes showed multiple micronuclei. It was striking because more than 50% of the tetrads showed at least 1 micronuclei, but frequently 2 or 3 as a result of problem with segregation. I also used fish fluorescing in situ hybridization to label some specific chromosomes and check whether they were always the same one involved in the formation of this micronuclei, but this wasn't the case. So how this translates in pollen viability and fertility? To check pollen viability, I used initially Alexander stain, which is stain viable pollen in purple and non-viable pollen in light blue, as you can see at the bottom in the CRISPR mutant indicated by an arrow. And this stain indicated that 28% of the pollen in the mutant was non-viable. Then I also used Folgen staining to check the developmental stage of the pollen at anthesis. So you can see the wild type on the top showing normal trinucleated pollen. And well, about half of the pollen grain in the mutant were not mature trinucleated pollen. So these apparently contradicting results suggest that the small pollen grains in the ZIP4 mutant were a mixture of both non-viable pollen, aborted pollen, and some immature pollen, non-functional pollen that didn't reach the trinucleated stage, and therefore it wasn't detected by the Alexander stain. So we decided to use the size of the pollen grain as an indirect evaluation of pollen fertility. And Abdul in our group came with the brilliant idea of developing a high-through pollen profiling method using a particle counter and sizer, which allows us to calculate very precisely the pollen grain size and the pollen number per anther. I won't explain the method, but if you are interested, you can check this published work where everything is explained in detail. So what you can see here is the pollen profile of 6 different wheat varieties, 2 tetraploid and 2 tetraploid and 4 hexaploid wheats. The diameter of the pollen grain is represented in the x-axis, and the pollen is represented in the pollen amount in the y-axis. And as you can see, all varieties show this typical normal distribution, with the mean around 50 microns of diameter in the case of the hexaploid varieties. However, when Abdul measured the ZIP4 mutants, and I'm showing here the Ph1 mutant and the CRISPR mutants, they showed a very consistent and similar pollen profile, which contained 2 very distinct peaks. So you can see the wild type in both cases in blue with a single peak, and the corresponding mutant in orange. The first peak in the mutants corresponding to pollen grain with similar size to wild type pollen, and the second peak with a considerable smaller size. So about half of the pollen grain in the ZIP4 mutant were smaller than in the wild type. And how this does it have an effect on seed set? This was a small experiment also performed by Abdul with between 20 and 35 plants per genotype, but the result was very clear. The grain number per spike was reduced by 40% in the CRISPR mutant compared with the wild type field, and about 42% in the Ph1b mutant compared to Chinese spring. So in short, the elimination of the duplicated ZIP4 copy resulted in more than half of myocytes with meiotic abnormalities, half of tetrads with micronuclei, almost half of smaller pollen grain, and over 40% reduction in grain set. So these results highlight something we didn't pay that much attention before, and it is the extraordinary value of the ZIP4 duplication on wheat fertility and grain number. But then, coming back again to this initial slide, although we have now better lines with only 1 gene ZIP4 mutated, these lines still accumulate a lot of chromosome rearrangement and eventually will become sterile, which complicates its use in breeding. But wouldn't it be great if we could uncouple these 2 effects of ZIP4? And this takes me to the last part of this talk and our more recent work, which kind of brings everything together. So I mentioned earlier that this duplicated ZIP4 copy performed 2 key functions during meiosis: promotion of homologous, promotion of synapses between homologs, and suppression of crossover between related chromosomes. And I have shown before that the CRISPR mutant had lost both functions, and the phenotype was the same as the Ph1b mutant. However, something was different with one of the ZIP4 mutants that I mentioned before, the one with the missense mutation. So the hybrids between this mutant and Viability showed the same level of crossover increases in the CRISPR and the Ph1b mutant, which indicate that it has lost the second effect. the ability to suppress crossover between related chromosomes. That was absolutely clear. However, there was a difference between the CRISPR and this TILIN mutant in wheat itself. So in the CRISPR mutant, about half of the myocytes showed abnormalities at metaphase 1. However, the TILIN mutant only showed 26% of abnormalities with no multivalent presence. All the abnormalities were due to the presence of univalents. And what it seems like a small phenotypic difference has actually very important implications, because what this suggests is that this TILIN mutant has retained the ability of ZIP4 to promote synapses between homologs, the first of the two functions. And if that is so, then this line represents a separation of function ZIP4 mutant. We have now renamed this line as the ZIP4 Ph1 the mutant, and further analyze its phenotype, confirming this separation of function, and with a very interesting result, as you will see for breeding. So first, I used GISH, genomic in situ hybridization, to check whether indeed there was no crossover between related chromosomes in this mutant. Because although there was no multivalent, it was possible that some of the bivalents were happening between related chromosomes. So here the A genome is labeled in magenta, the B in gray, and the D in green. And as you can see, all bivalents are formed between homologs, between chromosomes from the same genome. Moreover, even when some univalents were present, they resulted from crossover failure between homologs, as you can see, here, indicated by an arrow. So this confirms that the ZIP4 Ph1 mutant retains the ability to promote synapses between homologs. Then, as we did with the CRISPR mutant, I checked the myocyte development until the tetrad stage, and I didn't observe any major problem with segregation. There was a small number of laggard chromosomes, but no splitting of sister chromatids or chromosome fragmentation, as happened in the CRISPR mutant. And as a result, this mutant was associated with just over 50% of, 12% of tetrads with micronuclei. A considerable lower number than the 50% observed in the CRISPR mutant. As for the final pollen, you can see here again the pollen profile of the CRISPR and the Ph1b mutant with the two characteristic peaks size. And this is the profile of the ZIP4 Ph1 mutant in orange compared with its wild type in blue. You barely see the orange because surprisingly the pollen profile of this mutant and the wild type were basically the same, with only one single peak around 15 micrometers of diameter, indicating that this separation of function mutant doesn't produce significant number of smaller polar grain. And as you can imagine, with this pollen profile, grain setting is not reduced at all in the ZIP4 Ph1 mutant, something we already expected since, as you can see in this picture, the spikes and filling of the spikes in these mutants just looks exactly the same in the Ph1 mutant and the wild type. And not only the grain setting, but also the mutant itself is perfectly normal and homogeneous, contrary to the full ZIP4 mutant, which not only showed reduced fertility, but also reduced vigor and morphological abnormalities as a consequence of the multiple reorganization. Therefore, the ZIP4 Ph1 mutant represents a separation of functions, ZIP4 mutants, which allow recombination between wheat and wild relative while still preserving the stability and fertility in wheat itself. And for that, we strongly recommend the use of this new mutant in wheat breeding strategy rather than previously described Ph1 mutants. Also, very briefly, I want to mention that it was suggested that Aegilops variabilis might be contributing to the induction of crossover between related chromosomes, as it happened with some wild species. So it was important for us to show that the ZIP4 Ph1 mutant still allows crossover between related chromosomes in wheat itself, where no wild relative was involved. So I produced several haploids of both the ZIP4 Ph1 mutant and the wild type cadena. cadenza. So you can see on the left the haploid of the mutant, perfectly normal with 7 chromosomes from each genome. In the middle is the meiotic metaphase 1 of the wild type haploid in cadenza, where no crossover is present. And on the right is the haploid of the ZIP4 Ph1 mutant, where there is a mean number of more than 6 crossover per cell, same level as reported from seers using the Ph1b deletion, which demonstrate again that this mutant allows recombination between related chromosomes. So to summarize everything I have been talking about, we have identified a single gene, the meiotic gene ZIP4 on 5p, as responsible for the Ph1 effect on recombination. This ZIP4 copy performs at least 2 key meiotic functions: promotion of synapses between homologous chromosomes early during meiosis, and suppression of crossover between related chromosomes. And finally, we have provided several mutant lines where only the ZIP4 copy is mutated instead of having the big chunk of DNA, including 1200 genes deleted. These lines are available at the seed stores at GIC for anybody who want to use them. And definitely, we recommend the use of the ZIP4 Ph1D in wheat breeding strategies, since contrary to the other mutants, this line is perfectly stable and doesn't present any fertility problems. And of course, there are still plenty of unanswered questions, and we are already working on some of them, such as what is the role of the other ZIP4 copies on group 3, which are the ZIP4 protein interacting partners. Of course, we will also investigate the effect of this single amino acid change in the ZIP4 Ph1Z mutant, which produce such an interesting phenotype. And particularly relevant for breeding, is there any genetic variability in the duplicated ZIP4 copy that we can exploit? And can we obtain new ZIP4 mutant with useful phenotypic variation? And finally, I just would like to thank everybody who contributed in this work. I think I have mentioned most of the people as I went through. Thanks also to Tracy, who is now working with the ZIP4 on group 3. And of course, a special thanks to Brian Moore for the opportunity to work together in this great story for, as I said before, quite a while now. And thanks to you for your attention. I'm happy to take any question you have.
Thank you very much, Azahara, for a very interesting story that spans my entire lifetime. In fact, I was looking at when the first study was made, and I'm kind of it's kind of shocking. But one of the things that's very evident is there's been a tremendous amount of advancements in the past 10, 15 years, and a lot of that has been because of the technologies and the genomic resources that have become available. So just to remind everyone, you can put your questions in the Q&A panel, and we'll try to get to as many of them as we can during the remaining time that we have. One of the questions that came up, and I know there was a paper this past year on Ph2 encoding the mismatch repair protein MSH7 3D, which inhibits the wheat homoeologous recombination. Do you see these as complementary, or are they antagonistic to each other? Or has it even been considered yet, really?
I mean, we don't know. That will be the short answer, is that we don't know, but I think there are many possibilities that they are connected. I mean, it's true that MSH7, Ph1-2, it's before in the recombination network, is before, because it's involved in the mismatch repair, and later comes ZIP4. But as I have already mentioned, ZIP4, it seems to be acting as a scaffold protein, and it's involved in many more processes that we thought before. So if you think that really ZIP4 is changing or is allowing the recombination to happen between related chromosomes, you would think that is also, I mean, there are many chances that is also affecting the mismatch repair system. So I think there are many chances that there would be a connection, and that's actually when I'm talking about finding the proteins that are interacting. I actually was also thinking about Ph1 too, because actually I think, I mean, I always thought that the effect, I mean, what if you would mutate both Ph2 and Ph1, you didn't have an increase in crossover compared with Ph1. So it was like, you know, the same crossover that you get with Ph1, you get it when you add Ph1 too. But then I remember that Heidi here in one of the seminar was mentioning that there was a difference between genotypes. So I only checked Chinese Spring. That's the only one that I checked, and it was the same. So that's really good news also, because it seems that you can't use both together. But yeah, I kind of think that there could be any connection. It would make sense, but we don't know.
Yeah. So in fact, it might be very interesting as we get more information regarding the different locus that, or the loci, we can actually go in and do some network analysis.
Exactly.
Which could be extremely helpful, I would think. So
we actually did some, not me, that was Abdul in our group. He did some co-expression network. The problem was that because of this ZIP4 copy on 5D behaved completely, he got some meiotic module, and the copies on group 3, they were all in this meiotic module, but the ZIP4 was completely, you know, with the rest of the genes. But the group 3 actually showed interaction with MSH7 and MSH2 as well. So two mismatch repair protein. I mean, this is, of course, this is a co-expression. It's all very, you know, we need to prove it, but it's interesting. There could be a connection there.
Yeah, very interesting. What's the level of expression of ZIP4 5B copy in the Ph1 mutant, as well as in the duplicate Ph1 and Ph1C? So I think this, if I go back, yeah.
So can you repeat it again? What is the expression in the Ph1B mutant, or the ZIP4 copy on 5B is 0. It's not expressed because it's eliminated, of course. Yeah. Same in the Ph1C, it is eliminated, so there is nothing. I show the graph, so it's completely, of course, eliminated. And there is it doesn't seem to be a compensation from the other copies. But that's also interesting, because at some point we thought there could be a compensation, but there is not much increase.
Ah, interesting.
Yeah.
Okay. Is there evidence to suggest the ZIP4-driven mechanism might exist in other monocots or even dicots?
I mean, ZIP4 is everywhere, in mammals, in yeast, in fungus. It's a very, very important, as I said, it's one of these proteins that is controlling the main crossover pathway in plants, and as I said, in animals and in yeast, for example. So yes, it's present in, I would say, all the species that go through meiosis. And as I said, it seems to be a scaffold protein, so it's kind of managing different connections.
So it plays the same role in all these other species as well?
No, definitely no. That's always the thing with all these meiotic genes. It's all like very conserved. All the genes are in all the species, the mechanisms too. But then there are different, you know, differences between the different species, which bring me again why we need to study weight, because, you know, there are different tweaks. So in yeast, for example, when you eliminate ZIP4, there is no synapses at all. No synapses and no crossover, no interference crossover. Mm-hmm. However, in Arabidopsis and barley, there is apparently no effect on synapses.
Interesting.
Also, they mentioned that there might be a delay on synapses, so it might be similar, but basically it's mostly on crossover. And in wheat, this particular copy, the duplicated copy involved in diploidization, seems to have an effect, a subtle effect on synapses, and definitely on crossover formation. The group 3, we think only is having an effect on crossover. It's basically like the ZIP4 in diploid species.
Yeah, interesting. So is it possible that there's some silencing going on, or? For example, in barley, is there a way to test that? I mean, particularly given that we have the ability with CRISPR now to try to knock things out. Is there silencing maybe going on that could have an impact here?
Do you mean that the ZIP4 copy, the new ZIP4, is silencing something? Or
it's silenced in barley, for example. Is there something else in the regulatory pathway that is impacting it in something like barley or Arabidopsis, for example?
Sorry, I don't get it. So what do you mean in Arabidopsis?
Yeah, so it's a question of why is it not having the same impact in barley and hops?
I see. Sorry.
Can
No,
you
I
knock
think
out something? I mean, there's some things in
I
the
see.
regulation pathway that's blocking
I
it?
see. I think we don't know. That will be the short answer. But I think it's probably more about the protein itself. When you compare the proteins, the group 3 proteins are more similar to the diploid copy of barley and even Arabidopsis than the 4 copy. So this 4 copy duplicated from 3B is actually more different to the 3B. I mean, the other 3 copies are very similar, but this one is more different. The first part of the protein is almost, I mean, it has plenty of, and the structure is also different. So I think it's something, it could be something in expression, absolutely, because it's different, not only the level, but also the pattern of expression. But I think there is also something in the proteins, because the protein it has the same domain, it has this similar structure, but it's quite different.
Right. So could natural variation in ZIP4 explain why some candidate wheat varieties are prone to problems with aneuploidy and therefore fail the DUS test?
The DU test?
Yes.
What is the DU test?
I have no idea on the DUS test.
Me neither.
Maybe Chris Burt asked this question, so maybe he will explain. I'm sure it's a breeding kind of question.
DU test.
Yeah, so just maybe ignore that part and just say.
Sorry, so what was the first part? Whether there is variation in...
Could natural variation in ZIP4 explain why some candidate wheat varieties are prone to problems with aneuploidy?
I think there could be. I think that is very interesting. I think there could be, and I mean, probably a very. I will try to summarize because there are many interesting things about it. For example, even me, normally I work with Chinese spring. I don't see many univalents. Mostly they are ring bivalents. Then I started to work with cadenza because of the tilling, and I can see more frequently univalents, no doubt, and rod bivalents. So there is so obviously, I mean, and these are the only 2 varieties that I have been using. And I also can see it in the pollen difference. And I think there are many chances that there are exactly differences in ZIP4 and how it's making these early synapses to work properly. Actually, also talking about this natural variation, we also checked, now that we have the sequence of the Watkin collection available, that was a tool in our group, as for the data to Simon, and basically we checked how many allelic variants there were available of ZIP4. And while from the group 3 copies, there were in total, there were 5 in the A, 10 in the B, and I think 15 in the D. In the ZIP4 copy on 5B, on Ph1, there were 62 different allelic variants. So it seems that there is definitely allelic variation. We don't know whether it has, this could be exploited for breeding or not, but there's different, I think that there is definitely allelic variation. And there are few more data that seems to support this. So yes, I think that's a very good point. Yeah.
Yeah, and I think as we get more and more diversity and get more and more genome sequences, we'll be able to identify and narrow that down much more as well.
Yeah.
So the next question is, I was wondering if the tilling line uncoupling the different effects of ZIP4 and back crossed to WT or to wheat cadenza, do you think there might be other background mutations contributing to that specific phenotype?
Okay, we of course we back crossed it 3 times. And at the moment when we got the line, we were also checking that, I think that was Cristobal who could help us to check which other background mutation and whether, and we checked actually specifically the 70 megabase region and whether there were meiotic gene, and there was nothing that jumped out. Also, really, I mean, as you probably know, the phenotype of the Ph1B is really, I mean, there are not many genes that can do that. So I don't think there's any. Plus, as I said, it has been cleaned, and now we have used it. It has like 7 or 8 generations. So I doubt there is any background mutation that can do that.
So I have a clarification from Chris Burt. Thank you, Chris.
Okay.
DUS is distinct uniform stability test, and it's a required test to get listed in as part of a national listing for seeds in Europe and the UK. I knew it was something to do with breeding, but
okay. Okay, so yeah, that's really definitely very interesting. I never thought about it, but yeah, that's remind me that the difference between Chinese spring and cadence could be related. Yeah.
Yeah, and what you would see in others as well. So
yeah,
in other varieties.
exactly. Yeah.
So as you look at the future in the minute that we have remaining, what are your next steps? What are you planning to do now?
Well, now we are checking the group 3 to see what it does, and also whether we find any variability that we can use. And then I'm doing some more cell biology experiments with antibodies against the different ZIP4 copy to try to see where they load, how they load, to try to understand better the mechanism. And of course, I would like also to explore this variation, this natural variation, and see whether we can use it. So these are the main things that I have in mind at the moment. Yeah.
So are you trying to take advantage of any of the single cell genomics that are becoming available?
That would be great. That would be, I've been thinking about it for so long, and exactly that would be ideally, because many of what we do exactly, we are, even the expression data is all with the whole AMP30. It's really difficult to separate things. So I absolutely would like to take advantage, and I've been thinking about it for quite a while, to try to label some of the meiotic protein and pull them down and use really individual things. Exactly. That will help so much, definitely.
Yeah. Well, it's an area that, we haven't really seen the advances in the agricultural species yet that we started to see in other, particularly in mammalian and biomedical research, particularly. So it's an area that I have a great deal of interest in, trying to bring more of the single activity towards to work in agriculture. So, well, thank you very, very much for a great presentation, and it's interesting to see how the story has evolved, both in terms of my lifetime and in my lifetime in genomics. So, but thank you very much, Azahara, and I want to thank everyone for participating and remind you, you can go back and watch the video on the YouTube channel if you want to review, and also we'll be posting additional information after the webinar with regards to questions we didn't get a chance to answer. So, but thanks everyone for participating, and we look forward to seeing everyone at our next event. So have a great day.
Thank you.